Good afternoon, everyone, or good evening. So I'm from Paris, and the Sorbonne University, and I will talk quickly about the response of soil microbial communities to water stress in agricultural systems. And I will show you 3 examples of what for me could be important for practical research outcomes in the future. So my first, if it's going well, yeah. My first cue will be to perhaps go more deeply on studying diverse intensity of climatic events. If we are going, for example, in drought reweighting cycles, here on temperate soils in France, next to Paris, then the impact could be very different on the soil communities and functions like mineralization, for example. On the left-hand side, we make experiments in microorganisms on the water content, but in low intensity variation. So we are, for example, going from 60 to 30% water content in soil. And while studying bacteria and fungi, for example, we saw mainly that the communities were still, the communities are responding not too much, and they are highly resilient and resistant with some few differences. Fungi, for example, are more responsive to fluctuation in water. And also in this low range, CO2 is directly depending on the water contents. But if we are going more on high intensity drought, for example, here on the right-hand side, we tested some high intensity drought and heavy rain that are casted for Paris region in the future. So we made fluctuations with drought from 2 weeks or 4 weeks, and then heavy rain in once or twice. When we are applying this kind of high intensity events, then the response is different. First, for functions. So we still have a functional resilience for mineralization. After the rewetting of a dry soil, the mineralization is coming back to normal, except that we have important fluxes, important flush, which are already known as birch effect, for example. But if we are making experiments on the midterm, let's say 6 months, then we are beginning to see tiny differences that will accumulate with time. Here on the center, we'll have some different bacteria. On the wet soil, normal control. Here on the center, it's in blue, it's short droughts and long droughts at the end. And there is some different responses of species. Sometimes species are not responding to the treatments. Sometimes they are okay with the control, but they are beginning to disappear with stress and drought and rewetting. And sometimes also some species are not okay with control, but they are gaining some visibility and biomass and perhaps activity when they are under stress. So some species are more adapted than others. Another, I have one, okay. Another thing on the right here is when we are looking at diversity of bacteria with time, and we are accumulating some events, drought, reweighting, there is no huge difference in function and mineralization of soil. However, we can see in 6 months a regular and slow erosion of bacterial and fungal diversity with time. So to respond to the question, will the soil will be different in the future with climatic change? There are some differences, but sometimes it's something very tiny and that will accumulate with time, and it's not so easy to respond to that questions. So intensity of stress and drought and rain, and also In this context, another thing which is important is how you manage soil. For example, here, still around Paris, we tested some different type of management. On the left-hand side, you have intensive management. On the right-hand side, you have sustainable management, which means here a permanent vegetal cover, which is more sustainable for carbon conservation in soil, for example, and soil stability. However, when we tested the impact of drought and heavy rain on those soils, and we asked what kind of system will be much more resistant or resilient in the future next to Paris, the response is the higher resistance and resilience will be on the intensive management soil on the left. We have lower fluctuation of population and also lower CO2 emission. Our hypothesis there is that the communities in the intensive management are perhaps more adapted to fluctuation in temperature and water because of the bare soil in winter and autumn. So this is about conversion and management of soil. And I will finish on the last slide, still talking about conversion, but of natural system. And we saw before a talk about Brazil. In Brazil and also in Africa, when I work a lot, there are some tropical savannas and Cerrado in Brazil. And with time, those savannas are converted in other stuff like forests, or they are converted in arable lands or for doing agriculture. And this is something very important for stability of those systems, but also for questions about nutrients and also nitrogen, for example. On the left side, you have a figure about what is a savanna. It's an equilibrium between trees on the right side and grasses on the left side. And what we observed in savannas is that those different plants have different impacts on the soil functioning. Under trees in savannas, often you have a stimulation of mineralization, a stimulation of nitrogen cycling and nitrification, for examples, through leaching water and organic matter and litter. But under the grasses, it is totally different. The grasses are closing the cyclings by inhibiting different functioning of nitrogen, for example, nitrification. And we, for example, on the grasses, made observations that the grasses are inhibiting those processes through root exudation. So you have two types of plants community and a different equilibrium, all these savannas. The thing is that when you are changing that to a system or another, you will change totally the equilibrium for nutrients. If you are making afforestation to stock carbon, then you will go on the right-hand side, and then you will perhaps stock carbon, but you will lose more nitrogen through denitrification and lixiviation. And if you are transforming the savanna in grasslands, for example, you will perhaps go on the left-hand side, and here you will perhaps keep more nitrogen and nutrients. All of this to say that this is also in a context of climate change. And what we tested is that all those processes are depending on water content, drought, and seasonality. Generally, we observe that, for example, nitrification on the right-hand side up, genes and biomass of the communities in soil are increased by water. But if we are making a comparison of the plants and the seasonality, which is on the right bottom there, then it's totally different. For example, there is no impact of seasonality and water content on the bare soil on the left, under the trees on the right, but there is a huge difference for the grasses. And you can see that the activity here of RNA or also the biomass is lower when you have water than in the dry season. Our hypothesis is that it's linked to the functioning of plants. When there is water, the plant is functioning, and then it stops, and it's inhibiting the microbial processes. And when it's dry, the plant have no exudation. So I think it's perhaps three things to be taken into account in the future. And I'm just finishing there by some few references on this subject. Thank you.
Wow, Jean-Christophe, such a beautiful work you did. I have a lot of questions. Wow, it's because
I what?
you put you. I will. I oh la la. I have to meet with you, and I have a lot of points that to clarify with you. There is a lot of connection with the other presentation. I see connection with Lindsay, with Flavia, with quite everyone that presented something in this meeting today.